honor to have Ajarn Krabson here, who has traveled to Full-Time Club. I've known Ajarn Cry for about 7 years now. I first met him when our company, AA, invited him to come and talk about CI. Normally, when we hear about CI in academic circles, what do they usually talk about? Is it expensive? How much does it cost these days? And what diseases do they usually talk about? And what else? It's always about the brain and the heart, everything. But that day, when I went to listen to Ajarn around October 2018, it was really strange. I'd never heard before that. There could be an innovation that checks DNA and can tell if someone will get a serious disease. If we know that, would selling CI be easy? It's easy, right? There's no record in the hospital either, but they're not sick yet. Ajarn has even upgraded to the point of seeing something related to COVID that can cause serious diseases. Yes, please go ahead.
Thank you very much. Thank you.
Who here has gotten the COVID vaccine? Okay, put your hands down first. Who hasn't gotten the vaccine? Oh wow, over there in the corner of the room. Is that the Ajarn's team?
None of the Ajarn's team got vaccinated.
Hold on, once we finish listening, we'll know why they didn't get vaccinated. The professor said we can see death.
If we see it, whether it's deep or even if the virus itself mutates into other forms, there will be some mutations that we can see, and then we ourselves, we don't realize it. Yeah, we can't see it, but the genome we can see. The genome is all the DNA combined on the 23 pairs of chromosomes, which they can see in advance. And if this DNA gets heavily infected and becomes severely damaged, a lot, we can see it. This is about to lead to. The next slide.
Let's have you remember first, how many pairs of DNA are there? Oh, good job. 23, that's right. Okay, and the genome that I want you to focus on, are there any special pairs in the genome or?
So, in our genome, there are a total of 23 pairs of chromosomes, and the 17th pair is the one we use as a marker.
Remember this important number, 17.
The area on chromosome 17 that we use is an important marker. There is a gene called Angel Genome 534, located at position 13.
Wow, so there's 17 and there's also a position 13.
There's 13.
Today, we're not teaching DNA testing, but just remember this, because when you talk to customers, having this information makes you sound credible, right? It does. Customers don't really know how many pairs of chromosomes there are. They don't know which chromosome pairs or which DNA pairs, right, Professor? Okay, so this is the work of Femto Lab. What is Femto Lab, Professor?
Femto Lab is a lab for DNA testing. The reason it's called Femto Lab, yes, it's because back when I was still a professor doing research, I would have students. So they would know to gradually dilute the DNA, making it more and more diluted until it reaches the femto level. Even then, we can still see the DNA we've amplified. What is the femto level? Everyone knows nano, right? Nano is 10 to the power of -9, but femto is 10 to the power of -15. That's what our lab can detect.
Next, we're going to encounter ICs at the femto level.
Yeah, right now we've only found
Nano, not femto. Nano is okay, but it's still tiny.
Yeah, the dilution process makes it more and more diluted, okay, so that we can check how much DNA we have that actually works, and we can still see it, decode it, and then read the results.
Okay, let me briefly go over the professor's background.
I graduated from Chula, a food tech.
Yes.
Food technology, please. Master's, PhD, biochemist, graduated from biochemistry, science faculty. Mahadol, yes. Then I was a professor at the Department of Biochemistry, Faculty of Medicine, Siriraj Hospital, Mahadol University for 25 years. In my 26th year, I moved to the Faculty of Public Health to set up the DNA lab. But at that time, I started opening my own lab, and it became too much, so I resigned. So I was at Mahidol for 26 years. 26 years. But before I left, I had already obtained 22 patents in collaboration with Mahidol University, and I also received funding from the National Innovation Agency for pioneering innovative research. It was seed funding, you could say, preparing to set up the lab.
I've been doing this for 10 years.
Actually, if you count from when I first opened the lab, this year marks the 19th year. But at first, we focused on genes related to cardiovascular diseases, for which we obtained a patent. At the same time, we also had a cancer lab running in parallel. So if you count up to now, it's been about over 30 years that I've been doing this. But specifically, when it comes to infections, we've been working on this for over 10 years. Before we started working on infections, we did DNA testing to identify drug targets for targeted therapies. There were professors, top 10 oncologists in Thailand, who ordered tests from us because we used blood samples instead of tissue samples. At that time, there were only 4 labs in the world using blood samples, and we were one of them. Yes, please.
Let me clarify a bit about what I said regarding testing to ensure the correct use of medication. So, targeted therapy means that before you can use the medication, they have to test that person's DNA first. It's kind of like a custom-made drug, almost like a tailored suit, but not quite. Basically, they check to see if certain DNA markers might not respond to the treatment.
So we
Would have to avoid that. That's one type of test. But in your case, Professor, it seems like you went for a test that, as you mentioned, is related to cancer. Infection. Do we understand what causes cancer? Everyone? Behavior, genetics, right? Yeah, but it's surprising to hear from you today, Professor, that infections can cause cancer.
discoveries have won Nobel Prizes. Infections lead to cancer.
Professor, you mentioned 20% of cancer cases are caused by infections, bacteria, parasites, protozoa, fungi, and viruses. How does that happen?
So when we do genetic testing to select medication, to put it simply, it's a comparison between 2 labs. Our lab uses blood samples, while the lab in America uses tissue samples. The doctor will send the tissue sample to America and then compare the results. It's called race ball genetic in America. While this process is going on, pharmaceutical companies are developing ready-made drugs. And if you use the drugs from these companies, you can buy them at a special, lower price. We used it for testing purposes, but in the end, our costs were high because our lab was a research lab, 100% R&D, which took a lot of time. So we had to make a change. But the reason we decided to change was because during our testing, we accidentally found bacteria and fungi in the DNA of cancer patients. At first, we even thought the foreign lab had made a mistake until we eventually realized that this was actually true because it started to. There was already a database that had decoded the pathogens,
The professor once told me that in the past, we used to think that cancer was just a mutation of cells, that's all. But it turns out, as he once explained to me, that people who have these pathogens in their DNA, meaning they've contaminated the DNA, these people tend to get more severe cancer, and it recurs more often than in people who don't have it. Right. So how do these bacteria, parasites, and viruses get into us, Professor?
It's from environmental contamination. Eating contaminated food, for example, eating raw fish, eating somtam,
PLARA.
crab PLARA, not clean, things like that, right? Your favorite, Professor. It's raw fermented fish. That's really dangerous. Does this include salmon, Professor? Yes, salmon.
Salmon too.
Number 1 is salmon.
Salmon too is not healthy, okay? So today we found that cancer doesn't just come from not getting enough sleep or working too hard. Those are reasons too. They're one. bacterial meat, trying to implement various virus policies and such. After that, it just goes away. It shouldn't stay in our DNA. But the professor has found that there are cases where people who have these kinds of pathogens in their DNA actually have more severe symptoms. And sometimes, even after they've recovered, it returns. We've seen this before, right? We've heard about cancer coming back. How often does it return every few years? 5 years, right? If someone survives 5 years, it still doesn't mean they're out of the woods. They still have to keep monitoring. But what's important is that some people have a recurrence even sooner because the cancer is gone, but their behavior doesn't change. They go back to eating salmon again, right? So behavior can actually cause certain pathogens to be added back into the DNA even more. Okay, every year there are about 2 million new cancer cases.
From infections. Worldwide.
Just from infections, right?
Worldwide, 2 million cases. But now, that number has increased. However, this data is still from previous years, from 2012.
2012, right? Okay.
And then deaths.
1.5 million cases.
That occurred.
From infections.
And these infections, they make treatment more complicated and harder to manage, and there will be a higher mortality rate. Higher than other types of cancer. But the good thing is that, well, it can be fixed, it can be prevented.
Can be fixed. This word is like, it's like seeing the light at the end of the tunnel, Professor. First, I thought once that's it.
Accept fate.
So I thought just go all out and eat whatever you want. Salmon and all. But no, once you know, you can actually fix it. Hold on, there will be.
An example of something that can be fixed. Now, for patients who are infected, right? Microbiome. Microbiome means tiny, tiny things, the DNA of tiny pathogens. But when it's all the DNA combined, that's what we call the genome. When all the DNA is combined, we call it the genome, so that's why they call it the microbiome. But if it's just the pathogen itself, they call it microbiota.
Right.
Biota.
So to sum up, the DNA of those things just now, like fungi, parasites, viruses, right, Professor, is called microbiota.
It's called micro.
When it gets into and interferes with the DNA, cancer treatments, no matter what method, won't work.
If we don't clear the pathogens, they'll keep coming back and new cells will form. It can create new cancer cells, you know. There are plenty of research articles that say pathogens can get inside cells and then they go in and battle with various mechanisms inside our cells, and then they destroy our DNA. The main gene responsible for clearing this up is P53, which we currently use as a marker. As soon as it encounters the pathogen, it's like a fight, like a fighter or a police officer. If there are a lot of pathogens, it can get overwhelmed, leading to mutations, and the pathogens can infiltrate. It infiltrates and inserts itself into the patient's DNA. As it increases more and more and the immune system weakens, this person won't survive because these cells will turn into cancer cells.
You can just remember P53, jot it down if you want. P53, is located on chromosome pair 17. It's on chromosome pair 17. Now, P53 has two functions. The first is that it helps us control growth.
Another
function is that it acts like a police officer. The police will patrol all over the body, and if they find any cell that looks abnormal or doesn't follow the rules, like a criminal, they'll signal for it to be corrected. What do they call it again? Attitude adjustment, that's what they call it. Or DNA repair. But if it can't be repaired, it'll be killed off right away. P53 will kill it. Normally, P53 is very strong. It looks round like this. But whenever it's attacked, it will break apart. And then it itself will be tricked too. The DNA of the pathogen will turn into a crooked cop. So, if at any point in our body the P53 becomes a crooked cop, just imagine, not only does it not catch the thief, it even opens the door for the thief. So, P53 is really important. P53 causes mutations in this case. Just a bit more, so cancer starting from the ovaries, right?
Ovarian cancer.
This is caused by mutations of TP53 the most, right? That's what I said.
Exactly, yes, that's right.
The more types of cancer where we find a lot of somatic mutations.
Hard to treat.
So, the type of cancer that's hard or easy to treat really depends on the mutation of TP53. The ones above are cancers like
Ovarian cancer and also esophageal cancer. Esophageal cancer, right? And another one I can't quite remember, it's blurry, also head and neck and neck.
So these are cancers in the head and neck area and the digestive tract, right?
Yeah.
These are the ones that have a higher chance of mutation, okay?
You'll find them.
Mutation?
Yes.
What professor?
This one, let me tell you. Research aimed at finding ways to deal with mutated P53, searching for drugs, developing drugs. This has never stopped. There are reports every year. This is a paper, the latest research article, saying that researchers are working on ways to develop drugs, looking for ways to develop medication.
Developing.
When they encounter P53 mutations.
Mutations.
So, to sum up.
Today, when doctors in the hospital treat us, do they check whether P53 is mutated?
No, they don't check.
Do they?
They have.
Is there a test for it?
Yes, there are tests. There might be a biopsy or a buccal cell test. If it's the buccal cell test, that's for genetic screening, meaning it's hereditary. And if they find it in a tissue sample, they'll only check one or two positions, R72P, and another one I can't remember. But P53 is huge, so that doesn't cover everything, even though P53 itself is. It's really the star of the show. It was even on the cover of Nature Journal, 3 times actually. So sometimes it gets missed. There was a patient not long ago who had uterine cancer. She got her genes tested at a hospital where she was being treated, and when she asked the doctor said that her P53 was fine, not mutated, and there was just a tiny spot, just one spot at PR72P. So I told her, there are tens of thousands of G, A, T, C, over 20,000 bases. Just checking this one spot doesn't really tell you anything. And this particular spot, well, if you have 2 alleles, one person might have R72P, another might have R72R, and another might have P72P. The P72P variant is linked to a higher risk of cancer, but this doesn't really mean much for the patient because it's what they call a common single nucleotide polymorphism. But if someone doesn't, people think, oh, I've already had my P53 tested. But actually, that's not the case at all. Not at all.
It's very limited. To sum up, in hospitals today, in current medical practice, they only do very basic preliminary testing just at a few specific spots.
Not the whole bank.
Now, the next question is, if this person has a P53 mutation and then their DNA gets inserted, but they don't get tested and just go straight to taking cancer medication, what happens then?
If it's a conventional, mainstream drug, there are papers and research studies supporting that if P53 is heavily mutated, the patient won't be able to respond to standard cancer treatments, and there are drug charts for that. It means that even if you use them, they just won't work. About 25% of cases are like this. Sometimes, if you use the drug, the patient might die right away. They call it, well, we use the term Russian roulette, like out of 4 people, 1 dies. It's a gamble, really just taking your chances. Then for these people, if they survive after a year or 2, the cancer might come back again, and they'll have to use another set of drugs, which might even include some of the previous ones mixed in. So they might go through that and then move on. But if they survive again, they'll go to the 3rd set of drugs. If it gets to the third set, this time it might be the end or something like that, which is really tough because the mistake was made from the start. We should have tested for P53. In the past, when we tested for P53, it was to identify people who couldn't respond to certain types of chemotherapy. This is a test that, well, we have to say a lot of people come to get checked for it because if they don't respond, it means we need to change the treatment. There are also doctors who are researchers who can really understand and work together. For example, there's one case. He graduated with first-class honors from Rama. We could really communicate, read Nobel Prize-winning research articles every year, and have meaningful discussions. Another doctor, a professor, is an oncologist, one of the top 10 oncologists in Thailand. This professor actually uses our data and helps us out when we encounter cases in stage 3 or stage 4. He helps pull patients through, helps them survive by combining his methods with ours.
Professor, I ask for the juniors. Suppose today we meet a client who has cancer.
And then
We want to get a doctor who can communicate with you and understands you. Could you recommend someone?
yes, I can.
Okay, so we can recommend that this person is able to discuss things at the bean level and really understands the topic.
We will select for you. And I also want to mention that our work has been reviewed by professors from NUS, the National University of Singapore, which is one of the top universities in the world. Last year, it was ranked 9th, and this year, it's ranked 8th. So, when it comes to being in the world's top 10, sometimes communication between top 10 universities or research collaborations between these top universities means we have to be selective. For example, this doctor, he graduated with first-class honors and a gold medal, things like that. We have some really impressive examples. For instance, there's an elderly lady, probably around 82 or 84 years old now, who's been with us 23 times already. The third time she came was probably in 2016, which at that point there was only one type of bacteria left, and that particular bacteria was the one causing the pus. Professor Naren treated this person who used to have a really hard time by following our data. We kept discussing the case all along, and now grandma is about 80. Well, probably not over 84, and she's still healthy. But whenever she sneaks off with her daughter to eat something she shouldn't, pus will come out again, and then we have medicine for her to apply just like that.
Great skill. It gets in easily, you know.
Yes, but you can get it out. Grandma has one left.
So what is this, Professor? What is it?
For us, we don't actually see the parasite itself. In our tests, we only see its DNA. Now, this patient is in stage 4 cancer, stage 4 from colon cancer, which has spread to the liver and then to the breast, and then
To the breast and then...
we ran the tests and found a lot of different things, but what really surprised us, something we never expected, was the roundworm parasite, Ascaris lumbricoides.
We didn't find the actual parasite, but its DNA was embedded in the DNA,
P53.
in the P53 gene.
All right, and then
Let me just clarify something from what you told me. So this one, and this one down here, this is what DNA looks like when you test it, right? And this thing here, what do you call it again?
we do this using their white blood cells.
Our white blood cells come from our parents, we inherit them. If we test them at any age, they'll look the same, right, Professor?
If they don't mutate, then
This is basically like a standard, a control. But do you know that this person, male or female, might...
person female.
A woman, right? After she developed cancer, when they checked her DNA, looked at the state of her DNA,
It was really damaged. But when they checked, this is the area where the roundworm DNA attaches.
you can see the pattern, right? Just by looking at it, you can tell. It's chaotic, it's disorganized, isn't it?
Professor,
Yes. This one.
this one is from a Chinese person who came to get tested at a clinic in Thailand.
As well.
She had ovarian cancer. We found a blood fluke called Schistosoma japonicum in her blood. This parasite is commonly found in raw fish, salmon, and it's also widespread in China. Now, when we test, there are a lot of Thai people as well. The appearance looks like this. If you want to know what the parasite looks like, you can just search for it in Thai on Wikipedia.
So once we get the genetic code of whatever has invaded, we search it in something like a DNA library, and then we find out that it's the DNA of this very thing, the blood fluke parasite. Then we keep searching to see where it's most prevalent, because it's common in the city. And the customers who come are Chinese people. So we might have some endemic strains, right? Just keep an eye on it. But right now, we do have them, actually. There are a whole lot. This is the control sample.
Sample clean.
When you checked, how did it look?
Good.
Yeah.
It will be messy like this one. This is another liver cancer patient. And the cancer spread from the liver to the pancreas. This person is an elderly adult, over 70 years old, and in good health. He played golf, and then one day, the doctor who works with us asked him to come in for a checkup, and what we found was that we discovered several types of parasites. In this case, we took and sent the images, the ones showing threadworms, roundworms, those roundworms. Yes, the ones we just saw in the picture, Ascaris lumbricoides. So for this person, he didn't make it in time. He passed away. Passed away? Yes, okay, he passed away. I used to tell this case to others. This case really left an impression on me. He had a mild infection. It was purely bacterial. But what we found, this particular organism, we never thought we'd encounter it in humans. It's a bacterium found in tobacco leaves. Yes, in tobacco leaves.
Tobacco leaves, are you still there? Or maybe you vape and then you might encounter some cost-saving brands.
RC, right? Varic.
Yes.
Now, once we find it and get the data, what we have to do is look for references to see if anyone has ever reported that this particular bacteria causes cancer.
Yeah, you keep searching, right? Search, find the bacteria, and summarize it.
No.
No, the professor is going to keep searching about this type of bacteria to see what it can cause, searching through medical resources.
And then
I found that this bacteria causes
lung cancer. It's actually the first report in the world, the first report, in a research article. So we can use this as a reference. But the first case was in the lungs, while the one we detected is in the monkey. What do they call it? The trachea. The trachea. The trachea. Okay.
So, did you continue researching to find out where this bacteria is found? Could you tell us a bit about that?
Because I happened to find another bacteria that comes together with it.
Right.
It's found in the mud of the Proto-Mek River. I think someone reported Proto-Mek and that tobacco thing. Yeah, tobacco is right there. Exactly. So it's really exciting that. Yeah, so these germs were found in this patient. And that's how we found out that the professor had been smoking since he was a teenager back when he was abroad in Virginia. Virginia, that is. This bacterium managed to get into and live inside the DNA of.
Inside the professor's DNA? When you smoke, the tobacco leaf burns, doesn't that kill bacteria?
We don't know. We don't understand it, but we can see it. Even if it dies, parts of its DNA remain. The bacterium might not survive, but its DNA can still be there.
His DNA can insert.
Yes, that's right.
Okay. This one.
All right. Now let's move on to the next topic.
Who here has had COVID and how many vaccine doses? Professor, please go ahead.
Thank you. So it's like
COVID and the NGV vaccine.
Yes, so with COVID, well, they say it happens because we've had COVID before and then recovered. Those who have recovered are called COVID survivors. And these people are at risk of getting all sorts of illnesses, which I think you all have probably heard about already. Also, there have been a lot of reports about vaccine injections, but they're just not on Google, they're on rumble.com. But in the lab, we actually found this evidence from the DNA we tested, which we got from dried saliva samples. For some people who were too sick to provide saliva, we collected blood samples instead. But mostly it's dried saliva because sometimes the samples are sent from America, France, New Zealand, Singapore, and so on. This can lead to long COVID because it depends on where it ends up in the body in different systems throughout the body. Whether it's the neurosystem, the heart, all of it, as shown in this diagram. This was published in Nature in a major journal, so there's no need to argue about it.
They've already proven it.
Collected the data, and done the statistics.
The serious consequences, the negative effects, are what they call lasting impact. Even after recovery, it still leaves damage. Right?
Now, what we found in the lab is just like this. These are all remnants of the COVID virus. All of them.
COVID.
Just now.
Since we started talking, we've mentioned bacteria and viruses, right? But right now, we're focusing on the COVID virus.
Which we didn't expect to find because the world's leading scientists have said that COVID, or SARS-CoV-2, can never integrate into human DNA. Like HIV, we were the first lab to find this, and we were shocked. We thought we must have done something wrong, so we repeated the experiment. Then, with our initial lab data, we presented it at TAT. 3 samples, all positive. This person had been tested for COVID at a major hospital lab, and the results matched. But that test was just a check for COVID in the body. What we found was DNA.
Found it a lot, not small.
23 out of 2.
100 people who came to get tested with us had COVID DNA integrated 23%.
It makes you imagine it's like AIDS.
Because it integrates itself.
Just like AIDS, because it integrates into our DNA, just like AIDS.
Like,
exactly the same way.
What is this, Professor? Is it a cancer case?
Intestine. This is a case of a type of colon cancer that's hard to cure, and the patient was being treated at a major hospital. Here, they received a vaccine. The 3rd shot was Pfizer, but we can't remember the first 2 doses. But we have all the data on hand, and when we tested, we found remnants of the Pfizer vaccine in their DNA.
You can even identify the brand?
Yes. We can identify the brand because the person who reported it told us the brand.
Because, as you told me, that person actually took vaccines from each brand and sequenced the DNA for each one. Right, Professor? Then they checked which one it matched with, and it matched Pfizer.
All right, now.
This is technology. This is term, a new word.
It's a new term. You can look it up on Google. Just type in cancer and COVID-19 comorbidity. It's an emerging cancer, which is hard to treat. You can't treat it using the standard clinical guidelines or conventional methods. If it still lingers, it means it will just stay like that. There's no way out, no solution. Some call it an emerging cancer. Doctors refer to it as emerging cancer, but our team prefers to call it simpler. Kun Kitty came up with the name, and I really like it. Actually, I love it. I can't help it, I'm getting old. So, we'd like to call it COVID cancer.
COVID cancer.
COVID cancer.
Is this COVID cancer on the list of serious diseases? No, it's not. But is cancer on the list? Yes, it is. The company won't bother checking what kind of cancer it is. They'll pay out for any type. Hurry up and do it. COVID cancer, many sources.
The most.
Many sources.
You'll see an example of just how serious it is in a moment, okay? Now, this page here shows statistics, right? You can check the database, look it up on Wikipedia, Worldometer, or even Wikipedia again. They'll report on the number of cases, the number of people infected, and the number of deaths from COVID. The confirmed number of deaths is around 7 million people. The latest data as of 2024 is 7 million people. And then confirmed dead, right? Those are the people who passed away. But there are also people who didn't die, and we call them
survivors.
We're the ones who survived. We call ourselves COVID survivors.
COVID survivors. Are we COVID survivors?
Yes, we are.
COVID survivors, according to the statistics in my chart here, are 675 million people worldwide. But global statistics also say that the number of deaths is 7 million people. It's like a disease that came to wipe out the world, right? So now, next graph.
The next graph is about, well, vaccines. How many vaccines have been used worldwide? It's 13.72 billion doses.
That's 13.72 billion doses, right? Even here, we've probably used tens of thousands of doses already. That's already several thousand doses. Okay.
So we need to prepare, take care of ourselves, and make adjustments.
So what does it say here? This slide.
This slide says, in short, there's the issue of the so-called Anguli vaccine.
Yes.
And the government has already compensated. People have filed claims, and they've admitted that it's real.
Deaths and disabilities occurred. Claims filed are for these.
500, 5 billion, 5,000 cases, 5 billion.
And the government has already paid out over 2 billion in compensation. Hundreds of thousands of patients remain.
Yes, which will require more money.
258 million.
258 million, which is an enormous amount of damage. The world as it is will be shown in this graph.
The world as it is.
Yes.
Symptoms?
Arm weakness.
Severe leg weakness, too. Some even died.
Dizziness, headache,
Face
blacking out, numbness,
tightness.
chest. These.
These are the effects from the vaccine. Okay.
Yes. And then there are those cases that we're interested in because right now we've seen that there are people who suddenly have a stroke or something like that.
It's happening a lot these days, so
we started looking into it.
yeah.
So, if it's a heart attack or a stroke.
Yeah.
So, they say it's twice as much as before, especially in people with blood group AB and A. This is our data that they used in Healthcare Asia. Healthcare Asia, actually. You can look it up on the internet using Professor Clay's name as well. And over 8 years, you can see everything. There's pressure and all sorts of things like that. 8 years, right? This year is the 8th year that we've had this. The graph at the top shows that if it's cancer that isn't caused by infection, the graph is clean, overlapping cleanly. This is the somatic mutation, and it overlaps cleanly cancer.
Not an infection.
Cancer isn't caused by infection. For this family, we did blood tests. Tested 2 or 3 people, no infection.
The infection down below is...
The one below is cancer caused by infection.
Yes, I like it this way.
This is the COVID-19 virus. It gets really bad when you get infected, so all the proteins get messed up and can't function. P53 protein.
In our body, this protein is like a source of energy, right? If it's everything.
It's everything. It does everything. It works for the body. So,
if the protein can't function, it means everything breaks down.
Then the cells die. They can't function.
All right, so just choose which one you want to be, like the lotus or the one below.
Because
Right now, when you go for a cancer checkup at the hospital, they won't tell you if you're the upper or lower type. The hospital can't see the lower type at all because they don't check for it. They only see it when symptoms appear. But that's how it is. If you have more coverage, you're lucky you get the tests. If your coverage is less, you have to get in line for chemo. Not many.
if you have this kind of mutation on P53 or whatever it's on, you'll be 1 in 4 or 1 in 5 who will have to die.
1 in 4 who die, that's 15%, you know.
There's a paper that reports this for patients. I'll provide the reference document. Yes,
Okay,
this one.
this is the most recent award. I heard you say that they only give this award if it's something new to the world.
Right, so if it's a repeat,
If someone else has done it and you use it, no award.
so it has to be.
You have to be the first, the very first in the world to get the award. Okay, so what is this, Professor? This one,
This is the method for collecting my sample.
I thought it was a ballot paper. It's not right, Professor? So what is it?
It's for collecting samples, blood, and then making it into dry blood. You just drop it and send it to us, this one.
Sample collection.
We then tested it on a lung cancer patient. We took dried blood and dried saliva.
And then we did.
The lab results come out and the graph looks kind of messy like this. And it's the same strain, a type of bacteria. And this one down here is the control, which is DNA from the cells. It could be from their white blood cells or from the cells inside their cheek. We extract it and it comes out clean like this because, well, it's been there since they were born.
Let me explain. The sample on the left is taken from blood, and the one on the right is from saliva, both from the same person at the same time. When we test and examine the DNA, the results show the same infection. So what I want to say today is that testing from saliva is also highly accurate.
Yes, and right now, when it comes to collecting saliva, since we use filter paper for testing, which is the method scientists like us use, when we send out the filter paper, if it's already at stage 4, sometimes it doesn't make it back in time, or even if it does, we can't extract the DNA or RNA because if someone touches it, the DNA, which are enzymes on your hands, will break down all the DNA and RNA, making it impossible for us to work. And what's important is that during the COVID period, it was really tough. We couldn't receive any samples at all, none at that time.
Everyone was spraying alcohol everywhere.
Spraying alcohol,
Spraying everywhere.
going at it.
Used it all up.
So we tried a lot of different things, but one thing we did was use cotton pads, the kind you use to clean your face, like cotton pads, to collect saliva, anything we could. There was a French brand, an American brand, and so on, even from New Zealand, things like that.
Once we got the cotton pads,
We drip saliva, collect it in a clean cup, then drip it onto the pad. Once dry, we take another cotton pad, sandwich them, and send it to us in an envelope. That's all. This actually won an innovation award, the only one of its kind globally. Sometimes
people get blood drawn, but with this, there's no need. You don't need any personnel.
not at all, because during the COVID outbreak, if patients were at home and wanted to use human genome data, they could just use ours right away.
Yes, this is a project that you received an award for back in 2018.
Up until the year.
2024, we're receiving awards.
This year, 2025, we also got one. So there are 3 awards for Thailand.
How long to do the lab work, Professor? Suppose you have a saliva sample, how long?
It takes about 2 or 3, well, 2 months, because everything we do is online. If the power goes out or the internet is down, we can't get any work done. But if it's a stage 4 patient, we'll rush the process. Our team won't sleep at all.
The people who come to see you, Professor, at stage 4 are in really bad shape.
Yeah.
They're in bad shape and have been through a lot. They've been exposed to things they shouldn't have, and some treatments that weren't right for them they've already received.
Exactly.
They're just completely wrecked, honestly. I recommend that customers who are already healthy and care about their health, love their kids and wives, should also come in for a checkup. This is an award.
This is the most recent one I just received.
Today, Professor, you have 3 case studies to share. How about the first one?
About the first case study, this is a case that has already done a passion project. She's a 37-year-old cancer patient who was just recently diagnosed, hasn't even started treatment yet, and she's not afraid either because in her family, her grandmother is ill and undergoing treatment, and an aunt used to be sick but has recovered. She's the youngest in the house, so she's not afraid. She came to get checked, and when we found COVID remnants, we told her we still don't fully understand. We don't have research yet. We asked her to help explain what it means to have COVID remnants. What's going on? What we do know is this can cause somatic mutations, which can make the cancer more severe. I'll get in touch again. If I find any information or resources that could help her, I'll reach out again. That's it for now. This is about cancer and COVID-19 comorbidity, which will be written as a short communication, and there will also be references provided. You can search for it using cancer and COVID-19. For COVID, you can look it up by the title above, but we didn't publish this one. We used it for a press release in the hospital. The second example is a patient with colon cancer that had spread to the lungs, and the colon had already been removed and chemotherapy had been given. Now, this particular chemo drug can't be used if the p53 gene is mutated, but at that time, the patient hadn't yet. The test hadn't been done yet. After starting treatment, things seemed to get better, but then about a month later, the cancerous mass started to come back. So the patient switched to using herbal remedies and dietary supplements. Then in May, they came to get tested with us at the beginning of the month. When we got the first round of lab results, I told them that, well, it's not staying in one place anymore. So they went for a PET scan and found
small nodules
in the lungs, and they had spread. So there was a need to go abroad. The first injection caused a fever, and then a month later, the second injection was tested, and the second injection was effective for 10 days before he died.
So, in summary, in this case, I don't know if my own p53 is already damaged.
And then
Went to get mainstream medicine, which means modern medicine that they didn't test for, just received it and it went away. Because in 2019, she had it, but in 2020, it came back quickly. Like I said, it's because they didn't fix the root cause, just treated the tumor. So the root cause was still misbehaving as before, which is why it came back quickly. Once it came back, this time they switched to alternative medicine. So you switched to herbal medicine, right? Started taking supplements. All right. In the end, the critical point in this case is that in May, we started to see it spreading, and after it spread, there was a need to travel to Paragon. So it was required, right? The vaccine, the visa, right? Got 2 shots. Only 10 days after the second shot, passed away.
So there was evidence, things exchanged back and forth, right? Okay, so now the question is, I started to get suspicious because we're not the only ones working like this. Other scientists globally are doing this work and have come across similar things, but they just can't get published. So we talk, share information. Sometimes they send it to me, we discuss it, and it ends up on earonbird.com. On the computer, on Substack or something like that. We go in and read, and wow, they found the same thing too. But the thing is, no one can get it published. And because it can't be published in the research community, they just won't count it as credible. In the scientific community, only published work is recognized. Yes, got it.
Because sufficient number.
Only then will it be accepted. Or if it can't be published, it has to be patented or something like that. This is what made us wonder. If there already remains and we inject the vaccine, does anything happen? Does it stimulate something or activate anything? Let's continue. This is what the bacteria infecting this patient look like. If it's bacteria, there are several types, but we're focusing on E. coli and Mycoplasma. Mycoplasma is the smallest type of bacteria, and E. coli is commonly found and is quite harmful. It can cause DNA to break apart. It's the smallest one found in patients. It just keeps happening. And the thing is, it doesn't have a cell membrane. Without a cell membrane, cancer drugs, which act on the cell membrane, can't work. So it makes the drugs ineffective. It becomes resistant to all cancer medications. Mycoplasma is also tiny, just a bit bigger than a virus. Plus, it destroys the cells that are at the front line of the body's immune system. It destroys the natural killer cells.
NK cells, yes.
And together with a mutated TP53, it causes the NK cells to be unable to multiply and their activity decreases. If you encounter these things, it gets even worse. Yeah, so that's why this person is in a serious condition. As soon as we saw it, we knew it was really severe. This is the parasite. We're presenting the parasite, the plasmodium that comes from mosquitoes, because this one suppresses the immune system. Sometimes plasmodium, the malaria parasite, can get into our DNA, suppressing our immune system. Once immunity is suppressed, it makes us more susceptible to other infections. Many papers discuss this. At first, I didn't believe it either, wondering how a mosquito bite could cause this. What exactly does plasmodium do? We've all been bitten by mosquitoes our whole lives. Another parasite I mentioned is the threadworm. Threadworms are very serious.
Where is it, Professor? I'm trying to...
It's in our intestines. And then at night, if kids often have it come out as little worm-like things, kind of like tiny commas. It makes it hard for them to sleep, and they get itchy. This needs to be cleared up. And if we take steroids or use steroids, it might cause a hyperinfection. That means it multiplies and keeps infecting over and over inside the patient's body. For example, cancer patients or COVID patients who have this parasite and happen to need certain medications that might contain steroids. Those people might not die from the original disease, but could die from hyperinfection caused by strongyloides, the threadworm.
It's like a driver. When you add steroids, it's like a boost, really.
Boost.
Turns into a super parasite, right?
So when you encounter patients like this, besides giving them deworming medicine, when they go to the bathroom, they should wear gloves, Lena.
The professor advised me to take deworming medicine every 6 months. But with raw fish, it's every week. All right, that's fine.
Let's continue. There's also this mold aspergillus that we keep encountering, and we find aflatoxin in this context.
Beans, noodles.
Right, but here it's beans, yes. But in this case, we found its DNA, so it's interesting, found the DNA in the patient.
It's present in the patient.
This means the patient's DNA has mutated. indicating cancer. The virus we found, the red one here,
Yeah.
this is SARS-CoV-2, which is COVID-19. Number 19, the one on top, this one right here, that's it.
Sunflower chest.
Right, Professor? Yes, it looks like a sunflower, you could say. And then, when we see this part, this is the factor that causes us to get infected by the virus. We can see everything, whatever we want to see. We can see it all. Our data, this is it.
See it in the DNA.
And then once we have the graph and we analyze it, the part that is the virus, it looks like this. It has this appearance, and it shows that the virus will have a graph paired with these letters here, and it tells us, yeah, it is what gene, what species, it tells us everything. And which lab did it, we know all of it. This is to confirm that what we found is correct. Then we send the report.
A third example.
The third example.
This one is the most exciting. Cancer spread to liver. Had cancer. Family history.
There been cancer.
Had COVID. Got 1 dose of AstraZeneca and 1 of Sinovac, right? Just like us. In July, during the 67 exam, stage 4 colon cancer was detected, having spread to the liver. Tumors had spread to spots. From July to October, treatment started immediately. You started medication right away. Anti-cancer drugs. It's cancer, lung cancer.
It spread.
By October or November, it came back already. Was that fast?
They didn't know about the P53 mutation. They didn't test for it with us.
More medicine, no matter how good, yields the same outcome, the same medication.
Exactly. And with targeted therapy, it gets even more complicated. For these cases, there's a comprehensive database to support everything. So, if anyone gets tested with us, and it turns out to be a new drug that hasn't been reported yet, just send us the name of the drug. We'll look it up in the drug database for you, and we can tell you whether that drug is appropriate to use or not.
In this case, they came to consult with Professor Frame at the lab table, so we gave them some advice. As for us, right? We'll use extracts instead, because they can't tolerate chemo drugs or targeted therapies anymore.
Yes, exactly.
That's right.
And then we use our cell treatment, Frame to iCell.
Which is a drug called Frame to iCell as well.
It's a cell treatment,
Cell treatment.
it's a tailor-made treatment. NK cells or natural killer cells are generally available, but ours are tailor-made specifically for
That individual.
that specific person, just for that individual. If there's any infection, we can't see it.
Right.
So the Frame toy cell is unique to our lab.
Is this person still around, Professor?
Right now, yes, still here. And we were actually the first lab able to clear the COVID remnants from their DNA.
Applause for the professor. They found and removed it.
With this work, you sometimes prepare solutions. You never know if you'll run into a new language, and if you can't do anything next. We checked, and they have both before and after forms.
Ask it.
Now, with the before, you'll see that. There are all sorts of germs everywhere, full of bacteria and viruses.
But if you check a second time,
Anywhere that's empty means that germ has already been eliminated. It's already gone, okay?
long, Astra,
For this person, it was about 2 months. But we were able to do the first test for them within a month and a half because we had to hurry. They were already in late stage 4. And then, regarding the different strains, if the second channel is clear, it means our method can remove it. That means we can clear that strain. And then the next channel, the most important one, is about other viruses, but let's talk about this one. Here, the COVID virus. See? On the left, the second channel on the left, that's the before, right? There are 3 pieces of COVID remnants here. Yes, there are 3 pieces of COVID remnants here, right here.
This one.
3 pieces, 3 pieces. But on the second time, it's completely clean.
There's no COVID DNA left.
So,
What we took in, we were able to get it out.
we got it out. There is a way to remove it, you know.
We fixed this by
As for the solution, we'll provide all the reference documents showing which ones we used, whether it's medicine, natural extracts, or other natural methods. Sometimes it's not an extract, but something else, whatever it may be. And one of them is the use of cell treatments. Well, I'll give you a yes.
there are recommended comments.
Recommendations and references will be provided. For example, if someone suggests using garlic, you might wonder its source. In this case, information about the garlic will be,
It could be contaminated, infected.
something like that, you know? So yes, all of this is included. And this data is really exciting, but it just so happens that I'm not working on extracts like Chula rumfa, for example.
Yeah.
Its extract actually causes cell death without relying on P53. Normally, P53 would cause the cell to die as well.
No police involved.
Yeah, exactly. So the killing could be done by like CSI, DSI, or whatever name you want to call it.
Good thing the teacher got angry first.
Chula,
Okay, guys.
such as colleges, those.
Michelin.
Now, right here, what does it do? It triggers a cell program, which is apoptosis through the mitochondria and kills those cancer cells. Wow, turmeric can do that too. So when we work, we have to gather all this information. How can we help people like this? Recently, we have discovered castcore, which is a natural extract derived from the castcore plant, a specific type of climbing vine. Break time equals 0.5s/. It's truly amazing, almost like a cancer medicine. It stimulates cells, activating NK cells and boosting the immune system. However, when using it, we must consider other medications the person is taking. It must not conflict. We can't use it long term because it's still a medicine. You can use it for about 3 to 6 months, but it must not interfere with the patient's other medications. All of this needs to be backed by research. Otherwise, mistakes can happen. That's basically how it is. The important database we use, the one that allows us to provide services right to the patient's doorstep, is something we've already set up. They collect saliva samples at home and mail them in. This database is the Human Genome Project, which is the blueprint of humanity. Normally, it's a big deal and requires research centers or hospitals, but with COVID, none of this could be done. These resources weren't used at all because patients couldn't go to clinics. Hospitals were closed, everything stopped. So we came up with this method, collect the samples and send them to us. Now, we can read the results using both the Human Genome Project database, which decodes human genetics, and the Human Microbiome Project database. But all of this, you know,
Let me summarize this page for you. The top part is the human genome, but the bottom part is the genome of those tiny microbes. These are used as the professor's database.
it focuses on the environment, emphasizes infections, everything seen here, and mutations on P53. You can see all of them. There's also a table showing which mutations link to which types of cancer. And why are they interested in this? First, from decoding the human genome, they found that, hey, sometimes there are strange things. When they decoded it, it turned out to be bacteria. The first bacterium they found in human DNA during the Human Genome Project was Bacillus. The reason they did this was because there was now information that people whose genetics were passed down in families showing a tendency for cancer. But then there's the question, why is it that some people have the genetic predisposition but are lucky enough not to get sick, while others with the same genetics do fall ill? For example, Chinese people living in China who have the breast cancer gene don't get sick, but when they move to Canada or America, they develop breast cancer. This led to a major study, which started with experiments on lab mice. They used lab mice and performed gene editing.
Breast cancer.
They put the breast cancer gene in
Mice.
from humans, inserted it into mice, and then divided the mice into.
3 groups.
The mice were divided into 3 groups, right?
Mice divided into 3 groups. First group, clean mice.
No.
No contamination at all. Nothing to stimulate them. The second group of mice were normal mice. They lived normal lives like us. The third group were mice exposed to germs, placed in a contaminated environment.
After gene editing, they were still kept in a dirty environment.
There were 3 groups of mice, the super clean group, like us, and another group kept in a dirty environment.
They were also genetically modified, with the breast cancer gene edited in.
All groups got breast cancer. Why, Professor?
Okay, let me clarify. The first and second groups had the human breast cancer gene edited into them. Then one group was raised in a clean cage, and another group was kept in a cage where they could be exposed to infection. As for the third group, they were just regular mice, ordinary mice from the same family, but not genetically modified with a human gene. They were raised normally, without any gene editing. And the result was that the group of mice with a human gene modification, when raised in dirty cages, developed breast cancer. But the genetically modified mice, those with the mutated human gene, that were raised in clean cages did not develop breast cancer. And this is a database that's already been completed, but we're still working on it. New data keeps coming in all the time. For example, COVID just got added not too long ago. It's a database that, yes, so this one took quite a bit of time to work on. Since 2008 to 2016, it's divided into 2 parts. One part is pathogens, shown in blue, that cause disease and resistance. The other part is non-disease causing ones, meaning they don't cause illness. These non-pathogenic ones live with us symbiotically. This includes things like probiotics we consume. So this is
The good ones.
yes, the good ones, the good ones. As for the ones on the left, the ones with cis,
Yeah.
all related to resistance, all the NCDs.
Heart disease.
Cancer, diabetes, stroke, vascular diseases, depression. They're all included here.
The organic world in the world of non-communicable diseases.
Having children and it's expensive, or having difficulty conceiving. All of that is included here. All of these, we can work on them. We can handle them all. But our lab, we only want to accept specifically if it's autoimmune, for example, like what disease is it? Lupus, which is a type of autoimmune disease. You'll find these pathogens as well. Okay, let's continue. So this is to say that our work uses the work of Nobel scientists that we are passionate about, mixing and matching it. This one, Thai people already know about it. Thai scientists working in this field will know about polymerase chain reaction, synthesizing DNA in a test tube, extracting DNA from blood or cancer tissue or whatever, and then amplifying it. From a single piece of DNA, it becomes millions of DNA copies. Yes, this person is a Nobel scientist. This person is also a Nobel scientist whom we admire because he won the Nobel Prize twice. The first time was for decoding proteins, and the second time for decoding DNA. It's called Sanger DNA sequencing. He is the one who made it possible to decode human DNA, the human genetic code, in the Human Genome Project using this technology, which took 13 years. But now, they're updated technologies. Now, decoding everything takes just 24 hours. But those 24-hour methods are kind of like ready-made kits. If you want to publish your results, you have to take the specific part you're interested in from that 24-hour process and do Sanger sequencing on it. Only then can you confirm the results and publish them. So once you've decoded the DNA, what does it actually mean? Because for DNA and genes to function, you need proteins, hormones, enzymes, and things like that. Yeah, and all of that comes from amino acids. So you have to figure out how these things are connected, how G, A, T, and C link up with the amino acids that become proteins. This work actually won 3 people the Nobel Prize. If you study this subject, you'll be fascinated and never want to sleep. Starting from the very first person who began this work, Corona, the person in the middle, it just so happens that this is in the textbook we studied and now teach from, so it was possible to do. What they did was, once they had the poly-U, the amino acid climbed up the lattice, making it the first amino acid we found in this table. If it's DNA, it would be TTT, but if it's single-stranded RNA that's going to be made into protein, it would be UU like that, and so on until it's complete. You have to use mathematics too, not just biochemistry. Now, once we have that data, we need to read the information from the graph, the one from Sanger, in order to get the G, AT, C, C, which correspond to the amino acids or whatever. We have to read the data from the graph, and there's software for that. The database will be it's called CBI, which is under the MIS of the United States. Scientists of any nationality can access it. Right now, it doesn't seem like they're charging for it yet. Maybe Trump still doesn't know about this. So now we have to sit and do this work. When we have late stage for patients, we don't get any rest. We sit in front of the computer, rain or shine. As long as internet is working, that's where we are. We read results and see, for example, TP53 gene before we tested genes because drugs require them. But now we do TP53 gene. And infections, it's facilitating. I don't know how to explain it. It might take a long time until we get the DNA out as a piece of DNA, as I just mentioned. This is DNA. Then from DNA, it turns into RNA. RNA is like DNA, but it's a single strand. And then it becomes...
I thought it was an eye test chart. Which thing did you find first?
So I made it really small so you could visualize it, you know? This work, we really have to thank the scientists from the Human Genome Project who sacrificed their time to do this for us, because it's really exhausting work.
It's useful.
There are 8 points that we think from our work at the beginning, you know.
I also became a medical assistant.
Yes, so?
Before you decide about the medication, just take a look at it first. The second point is that this is the only lab that can use dry saliva, which is really convenient. And it can be tested both before and after getting sick. But most people who come to see me, are they the first type or the latter?
The latter.
Most people come to us after they're sick, rushing to buy insurance. Can you sell it? You can only sell BA, nothing else.
Testing.
In every organ, even the brain.
If you can't do it, just send it to us for testing.
Right?
Whether it's the lung, pancreas, or whatever, just send it to us. We can test it all.
We can test for all kinds of infections, whether it's bacteria, parasites, or viruses embedded in human DNA. And number 6, this is the first place where you can see the assets, COVID-19 in human DNA.
Other people can only see cultured human cells, but we are the only lab in the world where the actual living person can still walk around.
We can actually see it. They're still walking around inside.
When searching for cancer patients who don't respond to medication, we can suggest that maybe the chemotherapy they're using might
not be effective.
Is it dangerous to eat a lot of protein, Professor?
You have to check your kidneys, check your liver, to see if it's dangerous. If it is, you'll be able to tell.
This isn't related to DNA, but if your kidneys aren't functioning well, whatever you eat will make your liver and kidneys work harder. No matter what it is. Yes, if
like in our case, sometimes we do the tests and then we do a blood test and find, for example, that the BUN level is low, that means the protein is low. So we can supplement with protein.
Does it have to be protein specifically?
We prefer to be plant-based, but for animals it might need to be real meat, like fish, for example, because they really need animal protein as well.
eat fish,
Eating fish is fine, but there's something you must not do.
raw fish. Don't eat raw fish. My question is, if someone who is sick donates blood and then someone else receives that blood, what happens? Professor,
Dr. Satyakai, what do you think? Can you answer this right here? Stand up and answer.
Chick. You've been thinking for a while, right? It means there has to be a significant amount of the pathogen present. It could slip right into our system.
Or during a medical procedure, when we use certain instruments, like during an endoscopy, we sometimes find things like capillaria, for example, because once you see it, you see everything. And this is actually a global phenomenon, nosocomial infections, which every hospital in the world is trying to find ways to solve.
Let me clarify a bit, just to expand on what Ajahn said. What was it? Noso?
Nosocomial infection.
That means infections acquired from medical procedures in hospitals. Prem Tinsalananda.
So, Kun Win, are you here? Dr. Vikram Win?
Welcome, Dr. Wing. Let's give a round of applause.
I have to mention first that these eye-cell heart cells are much more special than the regular NK cells sold on the market, because the professor will first screen the patient's infection. They're very cautious, so they check to see if any of the pathogens we saw earlier are present. Once they've identified the pathogens, the professor will analyze exactly which substances need to be added to prevent and eliminate those pathogens. After that, the professor will combine them with NK cells. Some people, I've seen this in other markets, say, oh, if you have it, just buy anything available on the market. There are a lot of interbrands. But I have to say, the interbrands on the market come in many varieties, and each brand has different properties and their effectiveness also varies. So, we shouldn't let ourselves get fooled.
Thank you. Thank you.